In this video, we will discuss about how someone can determine whether a particular kind of cancers, whether a patient that has a pancreatic cancers will be benefited from this AOH1996 treatment or not. So this is the big goal about these uh, results that are presented here, and we will discuss a small portion or the approach that one can follow. So in this case, the approach that they have followed is that they check the organoid of the different cell lines and check whether they are sensitive to the AOH1996 or not, and thereafter they did the gene transcriptomic analysis and to find out what are the characteristics or what are the gene characteristics of those cell lines that makes them sensitive towards this AOH1996 and cell lines that are resistant towards this AOH1996. So the organoids that are tested for the different kind of cell lines are shown here, and in this case what they are able to find out that the cell line organoids from these cell lines, that is SU2C AJ, SU2C043 and JHH224 are sensitive. That is their viabilities goes down uh, significantly at a particular AOH 196 micromolar concentrations compared to the other cell lines which took a longer amount of this AOH, a longer concentration of this AOH 106 for its viability to goes down. And thereafter they plotted this they did this PCA plot analysis of the gene transcriptome analysis, and they're able to find out that there are some common things, or the sensitive cell lines aligns themselves in a particular particular area of this PCA plot, and whereas resistant cell lines align themselves in another area of the PCA plot. Thereafter they did the gene transcriptome analysis, and they're able to find out that in the case of resistant cell lines there are some genes that are commonly upregulated and commonly upregulated, and in the case of the sensitive cell lines there are other kind of genes that are commonly upregulated. When they did their analysis, uh, gene transcriptomic analysis involved what they are able to categorize these cell lines in different they are able to categorize these cell lines based on the markers, gene markers that are differentially regulated in the sensitive and resistant cell lines, and the categories are like basal like cell lines, classical cell lines, and these replication cell lines. Replication cell lines are the most sensitive to the AOH1996 as they will have more replication stress, and these are the markers that are considered for this replication stress in the green color. The markers for the classical cell lines are shown in the middle section. The markers for the basal like cell lines are shown here. The one marker for the classical cell lines is at the top, that is SLC40A1. And then they check the compare the IC50 values of the different organoids or different organoids from the different cell lines that are used in these studies, and they are able to find out that IC50 values are lower for the cell lines that are sensitive towards the AOH1996 as we can expect, and since they are sensitive they will require less amount of AOH1996 to reduce its viability by 50%, whereas the sensitive resistant cell lines has the high IC50 values, and this all in together we can see that the replication score seems to be higher in two of the sensitive cell lines, but it's lower in one cell lines. So this in all together this concludes that it's not always necessary that replication stress needs to be higher when that will makes them sensitive towards this AOH1996. So basically they do not make any concrete conclusions from these results, but the results is more inclined towards the case where if the cell lines has the replication stress, then they are more prone towards this AOH1996 sensitivity. But if they does not have the replication stress, then they might be sensitive towards AOH1996, or they might not be sensitive towards AOH1996. So I hope you understood their approach, and what are their main goals and what they are finally find out. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.